welcome to the California Botanical Society's monthly seminar featuring early career botanists, in this case, one who does not yet have his PhD, but is well on the way. So Tadeo Ramirez Parada joins us originally from Chile. He got his bachelor's at Tulane working with Jordan Karubian, studying drivers of biodiversity. And Tadeo worked on a tropical palm, and they were looking at sources of, in this case, phenological variation. And then he continued to work in the lab after he got his BS until he started his PhD at UCSB with Susan Mazur, where they're working, again, on factors affecting phenological traits using herbarium specimen data, field data, and computer simulations. He has not yet finished, but he already has 5 peer-reviewed publications and 4 preprints and other publications. As some of you who've come to these before know, I like to ask our speakers a little to tell me something else about what makes them happy and what they like to do, aside from their research. And so Tadeo makes music in his home studio that he set up. He records voice, guitar, bass, and synthesizer tracks. And when I asked him what genre his music would be, he said it falls into the indie rock or shoegaze categories. And for us boomers, for whom the term shoegaze is new, Wikipedia says it is a subgenre of indie and alternative rock characterized by its ethereal mixture of obscured vocals, guitar distortion and effects, feedback, and overwhelming volume. Tadeo also occupies his time and his mind, as he put it, delving deeper into the horrors and thrills of American history, especially through political biographies, which he finds a good way to gain an understanding of a historical period through a balanced treatment of broad historical processes and relatable individual level perspectives. So I think that all adds up to a pretty interesting person. And I think we're going to hear some very interesting research as well. So Tadeo, would you like to take it away?
Wow, what an introduction. Thank you so much. I appreciate it. And I appreciate all of you for, coming on a Thursday night to talk. I commend your stamina and I really appreciate your time. So thank you so much for joining me today. So I'll go ahead and share my screen. Can you see the main slide correctly? Yeah, so today I'm going to share some of the work that I've done for my dissertation at UCSB. Specifically, I'm going to share results from a study that finds evidence that plasticity and not adaptation, it might be the primary driver of temperature-mediated variation in flowering phenology in North America. And before I delve into it, I wanted to acknowledge, first of all, my co-authors that have collaborated on this paper, starting with my advisor at UCSB, Susan Mazur, and our close collaborator, Isaac Park, both of whom have been essential for carrying out this work, and also our amazing collaborators in the group, Sidney Record, Charles Davis, and Aaron Ellison. Each one of them has left its imprint on the project. And I also wanted to give a quick shout out to my dissertation committee, including Lee Anderegg and Elsa Cleland, both amazing scientists that I have seen a couple of versions of this project already and gave me amazing pointers in finding ways to communicate it and to polish it. So this is an amazing group of people that is responsible for the work that you're going to see today as well. So the very innovative outline of my presentation is going to be just to walk you through the background, just that covers the knowledge gaps and sort of conceptual cornerstones that motivate both the questions and the methods that I use to address them within this work, and to briefly overview the range of data sources and analysis that I did to uncover the main results that I'm going to show you and that we're going to discuss later on in this talk. So with no further ado, I'm going to start the presentation, starting with the background. So phenology, as most of you know, is the timing of developmental events throughout the life cycle of an individual or an organism. And it's an important, if not key, determinant of reproductive success. So phenology determines basically the biotic and abiotic conditions to which sensitive tissues or sensitive life stages throughout an organism's lives are exposed, and accordingly can mediate factors that range from the risks of exposure to adverse climatic conditions like frost or drought, or even determine the proportion of the growing season that might be available for an individual to grow and reproduce. But in the case of plants, phenology is not only important to determine reproductive success of individuals, it also defines the temporal availability of tissues upon which a wide array of organisms ultimately depend for growth and reproduction. And this can include herbivores, pollinators, or frugivores, just to name a few. So as such, understanding the patterns and the drivers of this phenological variation in plants is essential to understand the structure and function of both terrestrial communities and ecosystems. So as humans have probably known from time immemorial, phenology varies widely both over space and over time. And this variation appears to be evidently linked with climate. So the temporal axis of this variation is nicely reflected in some of the oldest phenological records around the world, such as this time series shown in the top figure of first flowering dates within a population of cherry trees in Kyoto, Japan, dating back to the 1500s. So the figure shows a time series in years in the x-axis, and the day of year in which the first flowers of the population bloom in that year on the y-axis, and the grade line shows the actual dates of first flowering in each year, whereas the solid one represents a smoothed line that just makes the trends over time a little bit more apparent and easier to read. And what we can see from that gray line is how sharp phenology varies from year to year within an individual population. The span of that variation from the earliest to latest dates spans about a month and a half, nearly 2 months. But that variation is also very apparent over space and has been widely documented across species and especially widespread ones. So for example, the bottom figure here shows the mean dates of first flowering for several populations of dandelion that are distributed across the former USSR. And each population here, each point in this graph represents a distinct population that was sampled. And it shows a sharp and mostly latitudinal pattern in which the southern populations flower up to 2.5 months earlier than the northern populations. So while it's clear that phenology varies along these two axes, it is unclear to what degree such variation reflects rapid organismal responses at the level of individuals or slower changes in genetic composition at the level of populations. So in other words, it's not clean across all contexts or clear to what degree spatiotemporal variation in phenology is driven by phenotypic plasticity, that is phenotypic responses to environmental change at the level of individuals, versus local adaptation, that is phenotypic variation within or among populations due to evolution by natural selection. So phenological variation over time is known to be probably predominantly caused by phenotypic plasticity. And work on model and agricultural species has uncovered the molecular and developmental basis through which the environment controls the timing of development. And through such work, it's been pretty clearly established that phenology is often predominantly driven by this plastic machinery that plants have evolved to synchronize development with suitable climatic conditions. But beyond agricultural and model systems, several observations have allowed us to establish how pervasive phenological variation over time is due to plasticity is among species. So for example, interannual variation often happens at timescales that are way too short to plausibly reflect microevolution. So revisiting the cherry trees in Kyoto, for example, the average lifespan of one of these trees ranges between 2 and 3 decades. But as you saw, phenology varies sharply from year to year, and therefore plasticity is the only plausible mechanism that could cause that variation. But even among short-lived species, phenology typically responds to climatic conditions that are experienced by adult plants within the same year that a phenophase occurs. So if phenological variation from year to year were driven by microevolution, we would expect associations between phenology and climate to show a time lag that is not often detected in wild populations. All elements that point towards the pervasiveness and even predominance of plasticity in driving phenological variation over time within populations and within individuals. But nonetheless, that said, local adaptation can generate variation in phenology over time, and because there can be evolutionary responses to climatic fluctuations, for instance. And microevolution of phenology in particular appears to be a very important process in facilitating biological invasions in North America. But nonetheless, such microevolutionary responses have been more sparsely documented. On the other hand, the causes of spatial variation in phenology are harder to discern. On the one hand, plasticity can generate such variation because the conditions that trigger a developmental stage often occur at different times across locations. But at the same time, different climate regimes over space can lead to locally adapted life history, including phenology. And as such, and there's been multiple examples that implicate plasticity or adaptation as the predominant drivers of spatial variation in phenology over space. But before going forward, why should we care about the drivers of, or the causes or the mechanisms or processes that drive this variation? And I think part of the answer is that as climate change unfolds, it is unclear whether plasticity might reach its limits. So revisiting the cherry tree example, you can see that after the onset of rapid warming trends depicted by the color bands, phenology has advanced substantially, and it's unclear, as it is with most populations, for how long this phenological advancement can match the warming trend. That is, we don't know if the plastic capacity of population will be exhausted as the climate continues to change. So assessing whether phenological variation across a broader spatial climatic gradient is caused by plasticity or adaptation might help us gauge the likelihood of phenological plasticity being exhausted. It can tell us both about the broader plastic capacity of a species and the likelihood that it will be exhausted in different parts of its range. The problem, however, is that assessments of that kind that partition plasticity and adaptation and its contributions have been historically limited because they typically require either resource-intensive or time-consuming experiments or extensive genetic sampling over space and time, which have restricted this type of analysis to a relatively narrow slice of the evolutionary tree. But fortunately, we've learned that under certain conditions, we can infer the relative roles of plasticity and adaptation from observational data. In particular, it was Phillimore and collaborators in Britain that assessing observational data from the common frog, learned that adaptation to spatial variation in climate can be inferred from differences between the slopes of phenology-climate relationships over space and time. We'll revisit that, so don't think that you need to get all the details right from that statement alone. So just to introduce some key concepts and vocabulary is something that we will encounter frequently is this notation of S space, which will denote the phenological sensitivity of a species to climatic variation over space. So in other words, it's the spatial slope of phenology, the relationship between phenology and climate. And this other term is going to be S time, which depicts or signifies the phenological sensitivity of a species to climatic variation from year to year or to interannual variation, which we will refer to often too as the temporal slope of the phenology-climate relationship. And the basic tenets of set of requirements for this framework to be viable, first is that S time, or the relationship between phenology and interannual climate variation, must reflect only plastic responses, which there will be scenarios where that assumption is not justified, but in many contexts it is. The second is that the relationship between phenology and climate variation over space, or S-space, reflects both the potential roles of plasticity and adaptation in generating variation along the gradient. And if those things are true, their difference, or S-space minus S-time, can be used to isolate the contributions of adaptation to phenological variation along the climate, the spatial climate gradient. So if these and other assumptions are met, we can infer the contribution of plasticity and adaptation to generating phenological variation along spatial climate gradients from empirical patterns of phenological sensitivity to climate over both space and time. And here specifically, and to mirror our analysis later on, we will examine how observed patterns of flowering time sensitivity to temperature over space, which we will call S space, and sensitivity to temperature over time, which we will call S time, can reveal the relative contributions of plasticity and adaptation along temperature gradients. So we will start by examining perhaps the simplest scenario in which individual plants do not show plastic phenological responses to temperature variation, but where populations of the species that occur at different locations along a temperature gradient flower at different times due to local adaptation. So in A, the x-axis shows temperature, whereas the y-axis shows flowering time. And the 4 orange lines depict how the flowering time of individual plants changes in response to interannual temperature variation due to plasticity within these 4 spatially segregated or separated populations along the temperature gradient. So the slope of those lines indicates the magnitude of the response. In this case, the flat lines would indicate that individuals do not respond plastically to temperature, for instance. So in turn, the dotted red line shows how flowering time varies among populations due to local adaptation along the temperature gradient. So in this scenario, local adaptation contributes to advanced flowering time among populations that are distributed in increasingly warmer locations across the gradient. So as in A, panel B shows temperature and flowering time in the x and y axis respectively, but instead depicts observed relationships between flowering and interannual temperature variation. So the slopes that we would obtain by correlating phenology to climate variation over both space and time. So in particular, the relationship between flowering and interannual temperature variation is depicted, or S time, is depicted by the orange line. And the relationship between the spatial temperature variation and phenology, specifically flowering time, is depicted by the dashed blue line. So the temporal slope shows how flowering time changes in response to cooler than average or warmer than average years, that is interannual temperature variation, given our assumptions and the sort of preconditions of this framework, we can assume that that response is caused by plasticity alone. That is an assumption of this framework. And in turn, the spatial slope shows how flowering time changes across increasingly warmer locations and is potentially caused by both plasticity and by local adaptation among populations distributed along that gradient. So in this particular scenario, as individuals show no phenological plasticity to temperature, as depicted in A, we should observe a non-significant temporal slope reflecting this lack of plastic responses. But in contrast, because the spatial slope can be generated by the effects of both plasticity and adaptation, and because populations, as we saw in A, are locally adapted along the gradient, we still should observe a significantly negative S space or spatial slope reflecting the sole contributions of local adaptation along the gradient. So the main sort of logic or component of our approach to estimating this, the relative contribution of plasticity and adaptation is basically going from B to A. So as the relative contribution of plasticity and adaptation should generate this empirical pattern provided that the assumptions of the framework are met. Observing this empirical pattern in turn implies this set of contributions of plasticity and adaptation. So we, by verifying that the assumptions of the framework are fulfilled by our data, observing B should imply that plasticity and adaptation contributed in this manner to generate the observed empirical pattern. But other relative contributions of plasticity and adaptation should generate different empirical patterns than the one shown here. So for instance, C shows a scenario where plants within populations flower earlier under warmer temperature, depicted by the negative slope of the orange lines, but in which there is no local adaptation among populations along the gradient, which is depicted by the flat red dotted line in C. So in this case, as the temporal slope reflects only plasticity, we should observe a significantly negative slope indicating earlier flowering in warmer than average years. But in turn, as the spatial slope reflects both plasticity and adaptation, we should observe a negative slope of equal magnitude to the temporal slope, and that is because only plasticity is contributing to generating the relationship between phenology and spatial temperature variation. Local adaptation does not contribute to producing variation along the spatial gradient, and therefore only plasticity operates and generates therefore a relationship of equal magnitude to that of the temporal slope. But plasticity and adaptation might act together to generate the spatial relationship as well. For instance, in the scenario shown in E, plasticity results in earlier flowering under warmer conditions, and local adaptation among populations results in earlier flowering in increasingly warmer locations along the gradient. So in other words, plasticity and adaptation operate concurrently in a co-gradient manner. That is, the type of phenological variation they generate under warmer conditions, it goes in the same direction. So in this case, the significant plastic response should lead to a significant spatial slope, as in F. But, sorry, one second. So in this case, the significant plastic slope should lead to a significant temporal slope, as shown in F. Which should be mirrored in the spatial slope as plasticity leads to earlier flowering in increasingly warmer locations along the gradient as well. However, local adaptation also contributes to generating earlier flowering across the gradient, adding to the effect of plasticity. And as such, it generates a relationship that is more negative or has a steeper slope than that would be generated by plasticity alone. So finally, local adaptation and plasticity might both operate along the gradient, but in opposite directions. So for instance, G shows a scenario where individuals flower earlier under warmer conditions because of plasticity, but where local adaptation among populations leads to flowering delays across increasingly warming location along the gradient in contrast. So in this counter gradient scenario, we should observe a negative temporal slope resulting from the effects of plasticity, depicted in the orange line in H. In turn, while plasticity would generate a negative slope, local adaptation would contribute to generating variation along the gradient in the opposite direction, which could lessen the amount of flowering variation along the gradient, and that in turn could result in either a shallower or less negative slope in this case, or even if the magnitude of the contribution is greater than that of plasticity across the gradient, could result in a spatial slope that has a different sign altogether. So as I mentioned before, the approach that we use in this paper relies on inferring plasticity and adaptation from the second row to the first row, basically. So provided that the assumptions of this method holds, these empirical patterns reflect distinct contributions of plasticity and adaptation. So by observing them, by measuring them and observing them in real world data, we can infer the processes that should have generated these observed patterns. So I've mentioned these mysterious assumptions a ton, but what are they? Well, just before I go on, I would just want to note that we've done a ton of analysis to assess whether these assumptions generally hold for our data. And obviously we can verify, we have thousands of species in our data. We can verify that all of them will hold for every single one, but we've done, I ask you to trust me just for this talk, that we've done as much as we can to verify that the general, these assumptions generally hold within our data so that the method is viable for the structure of our data set. So the first such assumption is that, as I mentioned earlier, the temporal slope, or S time, should reflect solely the effects of plasticity. The second is that S time cannot vary throughout the temperature gradient. That is, the responsiveness of flowering time to interannual temperature variation cannot differ among populations that are distributed along the temperature gradient. So in other words, plasticity needs to be uniform across the range of populations sampled. And the other one is that S-space needs to reflect phenological variation among populations along the gradient. So therefore, it needs to span a spatial scale that could plausibly represent populations, so that variation along the spatial gradient could plausibly reflect local adaptation among populations of the species. And finally, space and time cannot be biased. We have to be able to estimate these quantities accurately to be sure that what the signal that we're detecting is caused by what we care about and not by some other type of statistical artifact or unmeasured key variable. So we've done some effort to assess the plausibility of all of these assumptions, and I have some extra slides that I can show you if we have time in the end or in the Q&A if you're curious about seeing what we've done along these lines. So one problem is that to do this with field observations is tricky because field observations of phenology across multiple populations and years aren't available for relatively few species. So even though this method allows you to use observational records to assess the contribution of plasticity and adaptation, there's not a ton of field data that we can use to do this at very broad taxonomic and geographic scales. So here's when herbarium data come to the rescue, because they are the most extensive geographically, taxonomically, and temporally extensive phenological resource available worldwide. So there's an estimated 390 million records worldwide, and we're lucky in the United States that we have the highest concentration of digitized collections. And these digitized records cover thousands of species collected over centuries and spanning most of the continental United States. So they really facilitate analysis at scales that are not achievable through other data sources. And herbarium specimens capture the status of individual plants at a given place and time. So their collection dates tell us that the structures in the specimen, say leaves, flowers, or fruits, were present at that place and that type of year. So the locations and dates that are presented in the specimen label can be used then to link climate data to them, and therefore enable analysis of the relationship between spatiotemporal variation in phenology and spatiotemporal variation in climate. And accordingly, specimens have increasingly been used to estimate relationships between phenology and climate. And we and other researchers have found that they generate pretty reliable estimates of phenology-climate relationship over both space and time. So for example, in a paper that Isaac, Susan, and I published last year, we found that estimates of flowering time sensitivity to temperature obtained from herbarium data tend to closely agree those obtained from field observations. And we found that to be the case both for sensitivity to geographic or spatial variation in climate, which is shown in B and is depicted by that sharp agreement in the one-to-one line, and also for flowering time sensitivity to temporal variation in temperature, which is depicted in C, and also by that agreement in one-to-one line and the high correlation coefficient. So having covered that, I can move to the objectives of this study, which are first to estimate the flowering time sensitivity to temperature over both space, which we will call S space again, and due to temperature variation among years, which we will call S time, across thousands of species distributed across the contiguous United States. And then we will use these estimates to infer the roles of plasticity and adaptation as sources of variation in flowering time along temperature gradients for each of these species using the framework that I showed you earlier on. And finally, we're going to evaluate how the contributions of these two processes, plasticity and adaptation, vary among species that flower at different times within the year or that occur in different biomes. These two factors have been shown to be associated to substantial differences in phenological sensitivity among species in other studies. But to our knowledge, no other study has been able to do an assessment like this across such a vast range of biomes and spatial scales. So moving on to the methods. First thing to show you is a very cool herbarium data that we got to use for this study, which consisted of an initial 1.9 million clean and georeference records of flowering specimens that were collected between 1896 and 2020. And those ranges we had to impose to match it with the spatial extended climate data later on. And from some simulation and some power analysis that we did based on simulations, we decided that we should filter the data to only include very well represented species that were well represented by 300 or more specimens in the data. So this criteria ended up landing us over 1 million specimens of flowering plants across over 1,600 species across the contiguous US. And for climate data, we were mostly interested in temperature-mediated phenological variations. So we use mean monthly temperature conditions, so I'm going to call T-mean from now on, but average over the 3-month period preceding the mean flowering date of each species. So the time within the year over which temperatures were measured for each species was different among them. And then we partition this T-mean variation to a spatial and an interannual component to be able to measure phenological sensitivity to both these axes of variation. So the spatial component, we labeled the T-mean normal, which consists of a long-term average of T-mean conditions observed in the site of collection of each specimen between 1896 and 2020. So the T-mean normal represents a long-term average whose differences among sites therefore depicts geographic differences in chronic temperature conditions across locations. On the other hand, the temporal component here that we label T-mean anomaly is a deviation or an anomaly from the normal or the long-term average in the year and site of collection. So positive values of the anomaly would depict years that were warmer than average, whereas negative values of the anomaly would depict years that were cooler than average relative to the long-term mean for that site and that year of collection. So by doing this, we learned that T-mean varied much more broadly over space and among years. So for instance, the median species in our data occupied a range of T-mean normal conditions of 12.5 degrees Celsius after removing outliers, whereas the median species experienced a range of T-mean anomaly conditions driven by interannual variation of 5 degrees Celsius, so less than half. But crucially, T-mean normal and T-mean anomaly were independent from one another. So that shows that these two, this partitioning scheme does isolate two independent axes of climatic variation that reassures that we can assess them both simultaneously within the same models without issues of collinearity or other statistical artifacts that may arise. So a good sign. So the next step was to generate species-specific estimates of sensitivity to temperature over both space and time, which we did from a varying intercept and varying slopes model that include flowering DOY of each specimen as the response, and T-mean normal and T-mean anomaly as predictors. And this model for each species generated a slope of T-mean of flowering DOY versus T-mean normal, which quantifies the sensitivity of flowering phenology to temperature variation over space that we will label as space from now on. And the slope of flowering DOY relative to T-mean anomalies, also temperature variation over among years, that we will denote by S time. And both these sensitivity estimates were measuring days per degree Celsius. So what negative values indicates advancement under warmer conditions and positive values indicate flowering delays under warmer conditions. And then we fitted this model in a Bayesian framework, which basically just allows us to estimate the difference between the two, which indicates apparent plus adaptation based on the framework that I showed you earlier, while accounting for estimation uncertainty in each one of these parameters. So the model estimated apparent plasticity, and we learned through this that the model estimated apparent plasticity and apparent adaptation with very similar levels of precision, which reassure us that the patterns that we find are not because we can estimate just one of the two with much more certainty than the other. So a good sign. And finally, we use these estimates of apparent plasticity and apparent adaptation to assess the degree to which they varied with the mean flowering date of a species and among species occurring in different biomes or among biomes due to differences in species composition. And in particular, for biomes, we use level 2 ecoregions because they balance wide coverage. They're broad enough to encompass many species, while they're granular enough to capture meaningful bioclimatic variation across the contiguous United States. So they were like a happy medium that allow us to meaningfully conduct these analysis. So moving to the results. So the results that I'm going to show you basically depict the relationship between spatial slopes or S-space, which and the temporal slopes of phenology and temperature, which we termed as time, among the over 1,600 species represented in our data. So recall that within each species, the value of S space versus S time can reveal the relative contributions to plasticity and adaptation. So for instance, S space in particular reflects the effects of both plasticity and adaptation, or either one, along the gradient, while S time, given the assumptions of our framework, reflects the effects solely of plasticity. So in this figure, the solid diagonal line is a one-to-one line, which basically depicts perfect agreement in the magnitude and direction of S time and S space. And in turn, the dashed lines separate the negative and positive portions of the range of these two quantities. So negative values for both indicate earlier flowering under warmer conditions, either warmer than average years or warmer or progressively warmer locations. And the negative values and the positive values, sorry, indicate later flowering under warmer conditions, either warmer than average years or increasingly warmer locations. So each point in this graph represents one of the 1,600 or so species in our data, and the curved line indicates the observed relationship between S space and S time among species. So some overall results that are shown in this relationship before we delve into identifying patterns of plasticity and adaptation is that S space and S time tended to agree both in direction and magnitude among species. So our model estimated actually that the correlation between these two parameters was highly positive and likely varied between 0.86 and 0.89 with 95% probability. And in fact, just fitting the one-to-one line to this relationship explains 52% of the variance in the data. So it's a pretty good correspondence between the degree of phenological responsiveness over space and time for the species. However, the curved line does show that many species deviated significantly from a one-to-one relationship, which, as we will see later on, likely reflects the contributions of adaptation in deviation from this expected one-to-one relationship. So in this graph, the green points depict species whose sensitivity patterns were consistent with plasticity as the sole cause of variation in flowering time along the temperature gradient. And 41% of the species in our data show this pattern. So if you recall from the diagram that we went over a while back, and which is shown again as an inset in the panel, plasticity as the sole driver of flowering time variation along the temperature gradient should result in spatial and temporal slopes of equal magnitude and direction, as plasticity is the sole mechanism involved in generating both of those slopes. So this is because the temporal slope reflects the effect of plasticity, and if plasticity is the only mechanism causing variation along the spatial temporal gradient, its magnitude should be the same as that of the temporal slope. So accordingly, points that are on or near the one-to-one line, but away from zero in both for both of these sensitivity estimates, tended to be categorized as consistent with plasticity as the sole driver of variation along the gradient. And again, 41% of species in our data were categorized as falling within this pattern. In turn, the pink points in this figure show species for which adaptation appeared to be the only driver of flowering time variation along the temperature gradient, which represented about 7% of species in our data. So as a reminder, in the figure instead shows, so as a reminder on the instead shows, we should observe a non-significant S time if there's no effects of plasticity, and as S time reflects only the effects of plasticity, and that's depicted in this flat orange line in the diagram that I showed you earlier. However, we should observe we should observe significantly greater S space or a steep S space if the contributions of local adaptation to flowering time variation along the temperature gradient are significant, as they should generate a non-zero relationship between spatial variation in temperature and flowering time along the gradient. And this is why these species tended to show S time that was close to zero, so close to the horizontal dashed line in the y-axis, indicating limited or no apparent plasticity. But S space estimates that were far from zero in each direction, so that's why towards the vertical dashed line, there weren't many pink points, corresponding to scenarios where only adaptation is a plausible driver of the observed variation in phenology along the spatial gradient. and only 7% of species in the data showed this pattern. So in turn, 38% of species showed sensitivities that were consistent with both plasticity and adaptation operating along the temperature gradient, which in the diagram that I showed you earlier were depicted by either blue, that are depicted here, by either by blue points corresponding to species showing co-gradient patterns of plasticity and adaptation, or by light red points that show species with counter-gradient patterns of plasticity and adaptation. So if you recall from the diagram and the inset figure shown there, in these patterns, plasticity generates a significant temporal slope because it does significantly contribute to generating the spatial relationship. But local adaptation contributes to generate a spatial slope, or a space, that differs significantly from the temporal slope that would result only from the contributions of plasticity. So in the co-gradient case, local adaptations contributes to make S space steeper than S time, which is why the points in blue here are further towards the negative range of S space than they are in S time, and that's why they deviate horizontally from the one-to-one line indicating perfect correspondence. In turn, in the counter gradient case, local adaptation contributes to make S space shallower or to change direction altogether. Which is why the points in red are either further towards 0 compared to their corresponding values of S time, or have a different sign altogether in some cases, as those that are shown to the right of the vertical dashed line. So among these, about nearly 3 times more species show patterns that were consistent with co-gradient plasticity and adaptation than with a counter-gradient pattern. And finally, we found about 14% of species showed patterns that were inconsistent with either process as causes of variation along the temperature gradient, because there were limited responsiveness to interaction variation, both over time and to geographic variation in temperature. So just to recall some of these main findings, we found that 79% of species show sensitivity patterns consistent with plasticity. That is obtained from the 41% only with plasticity and 38% of both of them operating concurrently. And in turn, only 45% showed patterns consistent with local adaptation as a driver of variation along the temperature gradient. And actually, a plurality of species, if you compare all of these different categories, a plurality of the species show patterns consistently with plasticity alone as the driver of variation along the temperature gradients. And interestingly, most species that showed patterns that were consistent with the role of adaptation concurrently showed patterns that were consistently with the role of plasticity. So that's why only a handful of species ended up showing patterns consistent solely with adaptation as the driver of variation. So you might see from the range of values in the X and Y axis that flowering time, the sensitivity of flowering time varies very widely among species, right? And an important question in current global change biology is the degree to which that variation is ecologically organized. That is, does it either depend on species traits, on the native biomes that species belong to, or the time of years in which the species are phenologically active? What are the ecological or biological correlates of this variation? And that's kind of the focus of the next set of analysis. So these next results actually provide a first approximation to this question based on 2 axes over which we know phenological sensitivity varies significantly. One of them is the mean flowering date of a species, like what its phenological niche is, or where it tends to be active, or when does it tend to flower within the year. And among biomes, so biomes or ecoregions tend to show significant differences in the degree to which their member species respond to different climatic factors. So to start with a single biome as an example, this graph shows how the magnitude and direction of apparent plasticity and adaptation vary with the mean flowering date of a species within Mediterranean California. So the x-axis shows the mean flowering date of a species, and the y-axis the value of either its apparent plasticity or apparent degree of local adaptation along the temperature gradient. So specifically, the dashed line represents the mean apparent plasticity that is expected among, the mean apparent adaptation that is expected among species with a given mean flowering date. Whereas the solid line shows the same, but for apparent plasticity. So what is the expected degree of apparent plasticity for species that flower at that time of the year, whose mean flowering date falls in that time of the year? So each point in this graph, which are all collapsed as segments along the x-axis, represent a separate species. This is an interspecific pattern. And what we find for this individual ecoregion, this one biome, is that we see a stable or intensifying pattern of flowering advances under warmer temperature, both due to local adaptation and plasticity during spring. So it tends to be either intensify in magnitude or remain stably negative. And then we see a steady decrease in responsiveness throughout the summer. So a pattern of decreasing responsiveness in both apparent adaptation and apparent plasticity. And eventually an inversion to generating delays under warmer conditions, both due to local adaptation and apparent plasticity. And interestingly, when looking across biomes, so moving from looking at one of them to all of the ones that we meaningfully capture in our data, the pattern of advances in spring, reduced responsiveness in summer, and delays in fall was remarkably consistent across very diverse regions that range from very warm environments like warm desert floras to marine west coast forests that are temperate rainforests, basically. So a very wide range of biomes showed very stable and consistent patterns of variation throughout among species flowering at different times within the year. But nonetheless, ecoregions did differ in the precise timing of when the inversion occurred, especially for apparent adaptations. In some cases, say in this warm desert, the transition occurred much later than in the Western Cordilleras, and in also the magnitude of how different the contribution of each of these processes was among species throughout the year. So saying with the Western Sierra Madre Piedmont, the two lines seem to be pretty close to one another all throughout the year, whereas in the Western Cordilleras, the difference is pretty huge, except for the very extremes of the season there. So to discuss the main points very briefly, because I recognize that I'm running out of time. The main takeaway is that we find evidence that plasticity is the primary cause of historical variation in flowering time along broad temperature gradients, and that adaptation appears common among species, but comparatively appears to play a limited role. And other studies had identified systems or individual species in which plasticity seemed to be the predominant driver of variation along climatic gradients. But at least for temperature, our study suggests that this might be the norm and not the exception among North American angiosperms. And both plasticity and adaptation vary sharply with the phenological niche of a species. And these results were remarkably consistent across ecoregions and support studies showing this transition has been shown in other systems and within individual biomes often. So they should support studies showing transitions from advances to delays in response to warming among species throughout the season. But we show that such patterns are consistent for thousands of species and very consistent across biomes in the United States too. So it contributes to highlight the generality of these patterns that had been demonstrated before, and that to our knowledge had not really been demonstrated for apparent adaptation. So on that note, our results agree with Delgado, with a paper work done by Delgado and colleagues in 2020, which found using a similar method, basically the same method, found similar transitions in plasticity and adaptation throughout the growing season, but in the phenology of diverse organisms like saprotrophs, secondary consumers, primary consumers, and primary producers. So the fact that we find agreement when looking only at plants and flowering suggests that maybe the queuing mechanisms for phenological events that occur in the same seasons are shared even among unrelated organisms and for phenophases that are completely developmentally independent or unrelated. So again, points to the generality of maybe highlights the generality of this pattern of variation in responsiveness for events in general that occur at different times throughout the season. So just to conclude quickly, some notes of caution that are important and reminders that are important in hearing and eventually reading this publication when and if it comes out. Is that our results assess flowering time variation only along temperature gradients. So we did not measure the total flowering time variation that was due to plasticity and adaptation. So it's possible that their importance might differ along other climatic gradients, such as, I don't know, precipitation or any other important climatic factor. The other is that our approach might not detect all patterns of phenological adaptations along a gradient. In fact, it only holds if the contributions of local adaptation are monotonic and reasonably approximated by a linear relationship along the gradient. So if adaptation happens very patchily across the gradient and only in some portions and not the other, it is possible that we would lose or not detect the contributions of adaptation in such cases. And finally, our approach, as broad and exciting as it is correlational. So it's going to be exciting to see whether molecular and experimental studies do replicate the findings that we outline correlationally here. And of course, there might be confounds that we could not account for. I could not show you everything that we did to like try to weed out some of the most likely confounds, but we did a fair amount, but you can never be sure. I feel like always a caveat with observational studies. And finally, and perhaps most importantly, is that our results do not imply that species will exhibit adaptive plastic responses to ongoing temperature trends. And that is because, on the one hand, climate change might render these historical reaction norms maladaptive as the climate regimes continue to change, and because plasticity might be exhausted at the edges of the temperature gradient. So even though we showed phenological variation along a very broad range of temperature conditions that is caused by plasticity, that doesn't seem that that doesn't imply that the same space for further phenological shifts will be available at all portions of the range. So that's an important caveat. So just to conclude, we find evidence of plasticity the primary driver of historical variation in flowering time along temperature gradients. And our results appear to be consistent with those of detailed empirical studies, despite using different taxa, data types, ways of analyzing the data, regions, biomes, and phenophases, which highlights the generality of these patterns and the growing utility of museum collections in understanding biological responses to global change. So with that, I thank you so much for your time and open up the floor for any questions that you may have. So again, thank you so much for your time. Appreciate you
all. Great. Wow, that was really a monumental effort there. Let's throw this open for questions. I see a couple of people have unmuted themselves. Andy Simpson, do you want to just say it out loud?
Sure. I would predict that so I was thinking that I would predict that adaptation would be more important relative to plasticity the less local topographic variability there is where a plant is living. Because if you have a lot of topographical variability, that means it's easy to disperse up or down an elevation gradient, which whereas if you're in a flat region, you may have to disperse quite a long way in order to get to somewhere where the climate is different to your particular adaptational regime. Did you see this? Is there any evidence for this when you looked at different biomes?
Yeah, so I mean, I think that what's most interesting about that is that topographical diversity should increase the likelihood that there will be isolation at short spatial scales. So it would facilitate the emergence of local adaptation if necessary, right? Because it could still be that the plastic response does generate enough phenological variation that there's still no local adaptation occurring. As to whether or not we observed it, let's check. I honestly cannot remember off the top of my head. So let's go to that slide to see. So here, so I don't know, biomes like the Western Cordilleras appear to show some of the greatest differences between the relative importance of apparent plasticity and adaptation, with apparent plasticity having a much greater contribution to variation. So it doesn't appear to be the case here, but at the same time, these are very broad. The spatial scale of this analysis is so broad that maybe that would be something that plays out at a different spatial grain. But that's an excellent point that makes me want to look into it a little bit deeper.
Peter, would you like to unmute yourself and ask your question?
Yes. Thank you for a very good explanation of a complicated but really interesting bunch of data. And hi, Susan. Anyway, so plasticity is such a slippery concept. And I was wondering if, in a sense, flowering at different times in response to temperature might be a lack of plasticity. That is, the plant is unable to maintain homeostasis. It does exactly the same thing, and it results in earlier flowering, but it could lower the fitness of the plant. I mean, it's sort of similar to saying maybe it's injurious rather than adaptive plasticity, which would kind of go along with the greater influence of adaptation at the extremes of the flowering season.
Yeah, no, that's a super excellent point, because just because we find variation that is attributable to plasticity does not imply that that response is adaptive. It just shows that it occurs, that there are gradients that we can identify in which one mechanism and not the other appears to contribute to the variation, but it doesn't say anything about the adaptive nature of that variation. I guess over space, though, since this is a process that involves timescales that are much longer than sort of within population phenological responses, you would imagine that if plasticity was maladaptive, that signal would be removed by local adaptation. That the phenological change that is maladaptive, that is born out of the plastic response, would disappear as their selection for, a phenological timing that is not that which is generated by the plastic response. But I can only speculate with this data about the nature of the adaptive or maladaptive nature of the relationships that we uncovered. But that's also a great point that gets my gears going as to what data sets we would need to have to be able to get at. Yeah.
And if I could, follow up on that just briefly, I wonder if one could look for plasticity in things that are related to flowering at a different time. So, for example, would you have a longer fruiting season? Would you mature your fruits more quickly if you're flowering later, for example? You don't have so much time, and that would be an adaptive type of plasticity to look at.
Typically, there is very strong correlations on the sensitivities of sequential phenophases. So I've seen work mostly with woody long-lived species where the timing of one phenophase is a very strong predictor of the timing of the other one. So the entire reproductive development seems to shift back and forth depending on environmental conditions. So that would suggest that is the case in many systems. And even in model systems like Arabidopsis, there's the rapid cycling variant. Has very early flowering, but it also has early emergence, early leaf flush. It produces flowers at a lower stature, produces fewer fruits, but quicker. So it does seem that this is just one of the components of pretty integrated life history strategies more broadly. But that sadly, it's hard to capture that over hyperdimensionality or with herbarium data, at least with the kind that we have at these scales. But I think that seems to be the case, and it's a fascinating thing to think about.
Let me get a couple of questions that are in the chat here. So there's one from Edith Reed. Availability of temperature data is often very limited in ecoregions, especially over long historical time periods. What confidence do you have in the match between weather station temperature data and locality of plant specimen collection?
Well, that's an excellent question. I mean, PRISM does have very extensive spatial coverage that goes back to I think 1896 actually, but the further back you go, the more heavily modeled and the more estimated that those are based on sparser data. I think that hopefully if the bias is not critically systematic over time, with so many species, even those that are most affected by any mismatches of actual conditions and those of these grid climate products would sort of even out. But it's very hard for me to just parse that out a priori. We know that different climate products, at least for the period following 1980 or so tend to agree fairly closely to one another. And the bulk of our data was collected in over the past 40 years or so. So at least we know that the bulk of our occurrences did happen when there was way better climate data than there was, I don't know, a century ago.
Great, excellent. Why don't you stop sharing your screen so we can sort of see people? Yeah. So, okay, there's another question from Andy Simpson. Did you do any comparison between herbs versus trees? I ask because phenological data, Neotoma database, demonstrates that tree populations can withstand intense climate change, e.g. younger Dryas, but I would expect that shorter-lived species wouldn't have been selected for plasticity the way that longer-lived species would, and we have less paleo data on herbs because the fossil record is biased in favor of trees.
Yeah, that's an amazing question. We did analysis looking only at long-lived woody species, and the pattern was the same that I showed you, even though in a sparser data set of about 300 species or so, we found the same relative proportion of these patterns. So at least we know that, in cases where the responses were unambiguously plastic, and even though we don't know the adaptive value of them, the pattern remained. And when we substituted to exclude those that were long-lived perennials, the pattern remained consistent. So at least we know that it's not a growth form and life history don't appear to substantially alter the correlational patterns. The adaptive nature of the patterns that we do find is something that this data cannot illuminate, and your comment, I think, kind of gets to that. I mean, if we could get more data to sort of parse if the relative frequency of adaptive plasticity differs among groups, that would be super cool to sort of like kind of add a dimension to these sort of broad kind of agnostic as to the adaptive nature of them analysis.
There's one question that I'll get you in Professor Mazur in a second, but there's a question that just got sent directly to me in the chat. Can you predict from your efforts which species will survive given climate temperature changes? Can you predict from your efforts which species will survive given climate temperature changes?
I don't think so, unfortunately. Both because what we show is a historical pattern. It's like how plasticity appears to have contributed to generating variation along spatial gradients that were established long ago. And so that's one thing. And because its historical reaction norms might become maladaptive as the climate continues to change. For instance, there are examples in which the environmental cues that are used to trigger development become decoupled with the environmental factors that determine the fitness consequences of reproducing at different times. So if, I don't know, phenology matters because it determines synchrony with pollinators, and then the phenologies of two species becomes decoupled because the adaptive value of the historical reaction norm might disappear, and the norm might become actually maladaptive. So these reaction norms do not predict what will and will not be adaptive in the future. I think that's, I mean, and even historically, we cannot know if, we were talking about at the beginning, if these correlations do highlight adaptive relationships to begin with. So that, I feel that that's a dimension that we cannot quite crack with this data yet, or I haven't thought of how I would go about trying to do something.
I think it's pretty wise. Susan, question?
Yeah, no, I was just want to respond a little bit to Andy's question. I wouldn't really agree that shorter-lived species aren't selected for plasticity. Just the traits and the time of which that plasticity is expressed may differ. So we know that germination times are highly plastic, and germination times have cascading effects on how big a rosette grows, how long it takes a rosette to get to a threshold size, and consequently what time flowering occurs. So this is a challenge because even with annuals, we can't assume that temporal variation in flowering time is a plastic response versus a microevolutionary one. And I'm not sure. I'm not sure what you were getting at with paleo data for herbs, because none of these results depend on paleo data.
Yeah, no, but the reason why I was bringing up paleo data is because we know from paleo data that trees are capable of withstanding fairly intense, fairly rapid climate change, but the paleo data don't tell us for herbs. So I was asking, what do you basically a version of the next question that got asked is, what do you think? Do you think you can say anything about how herbs are going to be able to withstand climate change relative to how trees are demonstrated to be able to withstand climate change?
Fair enough.
Great seeing you, by the way.
Likewise.
Al Mackey, would you like to ask your questions? You're sending them directly to me, and it's not my work, so I can't answer them. Apparently not. Okay. I'll read it to you. The first question says, in response to whether you can predict climate change, well, what will survive climate change, he says, so of what value is your work then? And then he follows it by saying, so can it be used for management practices? I started to write a lengthy reply, but I thought I would let you do it instead.
No, that's an excellent question. I think everybody is kind of compelled to give an answer to that question with anything we do. I would say that the path to figuring out what will survive or not climate change is so complicated, and it requires so many pieces of information. And a very early piece of information that you might ask is like, well, what is the apparent range of plasticity of species in general? Like beyond the few in which we have like good experimental data and good molecular data that we've quantified, what in general seems to be the range of variation that plasticity can generate to begin with? Seems like a preliminary question to then figuring out what is the adaptive value of that apparent plasticity. So I think this work is useful in outlining a very general pattern of the range of variation that apparently can be caused by plastic responses, to then figure out ways in which we can sort of get deeper into assessing the adaptive value of such responses. So to me, it's a first step in that direction, and of interest. Just kind of on my nerdy side, it's just that I'm like genuinely interested in understanding the drivers of trait variation in space and time. So that's kind of like in the basic science side, but from a practical perspective, I think it's a first step towards achieving that degree of prediction that is very hard given like the complexity of ecological systems. So, a grain of sand.
Yeah, you got to start someplace. Isaac, would you like to?
Well, I just wanted to chime in on that another piece of this is that if phenological variation is carried primarily through adaptation versus primarily through plasticity, even though, as Tadeo mentioned, there's a possibility that you saturate and reach the edge of the range through which plastic responses can occur, the implications for when a plant's timing is going to be 20 years from now or under 1 degree Celsius additional warming or whatever are very different if we're talking about the variation that we see being carried adaptively versus plastically, because that would mean it would take longer time scales to respond. So this work doesn't necessarily directly point us to what is adaptive, because there's all kinds of things that can be happening. Like Tadeo mentioned, you could decouple synchronies, or you could be at greater risk of frost damage, or all drought stress, all kinds of things. But what it does do is it puts us in a better position to predict what the timings will be, which, if you're not separating out plasticity and adaptation, you could get it wrong much more easily looking at historical patterns.
Everything helps. Every piece of information helps. Plus, personally, I'm a believer in basic knowledge, basic understanding of what's going on out there. So, okay, any other questions before I fade into darkness here? Oh. Well, thank you very much, Tadeo. That was really, really informative and really a monumental study. Yeah, Thank you very much. And when do you think you're going to finish?
Well, first, thank you all so much for coming again Thursday night. It's heroic that you're all here, and I really do appreciate all of your questions and your attention. So thank you so much. As to finishing, ooh, we'll see. I think hopefully within a year. I think I'm well positioned, but you never know.
Excellent. Excellent. Well, I look forward to hearing more from you in the future. Yes, and see whoever in Boise. Take care, everybody.
Bye all. Thank you so
Bye-bye.
much.
Bye, everyone.